Good evening, everyone. My name is Louise McCarthy. I'm the Director of Alumni Relations at Monash University. I'm really delighted to welcome you here to the final event in our Alumni Speaker Series. Could I please ask you to turn off or silence your mobile phones? To begin tonight, I would like to acknowledge and pay my respects to the people of the Kulin Nations as the traditional owners of the land on which we meet. In particular, the Wurundjeri and the Bunurong people, and to pay my respects to their elders, past and present. I'm delighted this year that we've been able to bring you 4 entertaining and unique events as part of our speaker series. We started the year discussing the role of the AFL in our society. The event was moderated by alumnus and star of the ABC's Gruen Transfer, Russell Howcroft, and included an illustrious panel of experts. We then launched the much celebrated history of Monash University, University Unlimited. Our third event was something very different to what we'd done in the past. World-renowned musician and composer Paul Grabowski candidly discussed his life and love of music and gave a spectacular original piano performance. I hope some of you are here to see it. This evening, we are here to talk research. Monash University has some of the best minds around. Our researchers are making an impact across more than 150 fields of study, including several Monash specialisations. Tonight, we will learn more about the breakthroughs they are making in the fields of ageing, health and wellbeing. Is there a way we can not only live longer, but live better? Joining us this evening are Professor Peter Curry, Dr. Erica Sloan and Professor Colette Browning. Professor Peter Curry is Deputy Director of the Australian Regenerative Medicine Institute and an expert in the field of zebrafish genetics, an exciting strand of regenerative medicine that could spark treatments for conditions such as Parkinson's, dementia and muscular dystrophy. Dr. Erica Sloan is a cancer biologist at Monash Institute of Pharmaceutical Sciences and a senior lecturer at the Faculty of Pharmacy and Pharmaceutical Sciences. Her research focuses on chronic stress and how it contributes to the development and progression of cancer. Professor Colette Browning is the Director of Primary Care Research in the Faculty of Medicine, Nursing and Health Sciences. Her research focuses on healthy ageing and improving the quality of life for older people, chronic disease self-management and consumer involvement in healthcare decision-making. Please join me to welcome our first speaker, Professor Colette Browning, to the stage.
Well, thank you for a lovely introduction. I'm very pleased to be here. I'm actually an alumni of university myself, and I've only returned in the last 5 years, so it gives me a lot of pleasure to be speaking today on behalf of the Alumni Society. As already mentioned, my interest is in what might be called healthy ageing. How can we actually improve the quality of life for our older citizens? So, as way of introduction, first I'm going to talk a little bit about population ageing, just to really set the scene for this topic we have today, and the associated health issues that are related to individuals as we age, because we are all ageing. And then I'm going to talk about, fairly briefly, just give you a bit of a snapshot of 3 studies that we've been doing over the last 4 or 5 years in the broad area of healthy ageing. So first I want to look at population ageing. So many of you may already know that at this time in history, the life expectancy for Australians is quite high. For example, if you look at the table that I have up at the moment, you can see that the life expectancy at birth more recently for women is around 83 years and for men around 78. And yes, women do still live longer than men, but unfortunately, even though women are living longer than men, they're often living with a number of chronic conditions, whereas men tend to become ill and then die. So I don't know which you would prefer. And if you compare that to the turn of the century, at birth, the life expectancy for women was only around 58.8 years and for men around 55 years. So you can see over that period of time, over 100 years, life expectancy has increased significantly. And I would say that this is a cause for celebration. We should be very proud of the fact that now our life expectancy is so high. So when we look at the population statistics in Australia, in 2011, the proportion of people over the age of 65, which is generally taken as the cut-off for old age, was 13.7%. However, what we're going to see over the next 40 years or so is a rapid increase in the proportion of people over this age. So by 2050, almost a quarter of our population will be aged 65 and over. And I think that's something to reflect on. How will our society look in 40 years' time with that sort of profile of older people? Perhaps then older people will actually rule and have a lot of influence on what governments do. Because ageing, however, is associated with the increasing likelihood of disease and disability, governments are often concerned about the fact that we have an ageing population. And you'll often read in policy documents about population ageing that it is a burden. Whereas I would like you to think that, as I said previously, it's really a triumph that we are able to live such long ages now, till such a late age now. And also, even though we do see increasing health problems as we age, from a social perspective, older people still contribute significantly to society. And you can just think about your own families from this perspective. Most of us, have grandparents who provide a lot of support within the family, both financially and with looking after children. Older people are very great volunteers. They contribute significantly to society. So I would like us to really not think about ageing as a burden, but as a triumph and something that we should celebrate. Now, just to put the context back again to the issues around health and ageing, even though I would like to for you to think of ageing as a positive experience, we still are struck by the fact that as we age, we're more likely to develop particularly chronic illnesses. And on this slide, I've just listed some of the major chronic illnesses that we see in the over 65 age group. And you can see that musculoskeletal conditions such as arthritis figure very prominently there, hypertension, high blood pressure, back problems, back pain, heart disease, right through to dementia, which the rate of dementia in the community of over 65s is about 9%, and this increases dramatically in the over 80-year-olds. So when we look at, the diseases associated with ageing, it can put forward a bit of a grim picture, but I'm hoping to maybe change your mind about that a little bit as we go on. One of the ways to look at this is that a lot of the diseases or health conditions that are associated with ageing are actually related to what we call risk factors. And they're risk factors that are what are called modifiable. They're risk factors that are changeable. So most of those conditions that I listed on the previous slide are actually highly related to risk factors such as being obese or overweight, being inactive, or having a poor diet. So if you look at that slide I've got up now, you can see that particularly overweight is becoming a significant problem in our middle-aged and older groups, where even in the 75 years and over age group, about 63% of our population would be considered overweight or obese. And inactivity is a big problem in older age groups. So even in the middle age group, in the 40s and early 50s, nearly 3/4 of the population don't exercise enough for a health benefit. But what we need to keep in mind that these risk factors are modifiable. We can do something about them. So in response to population and in response to the fact that a lot of the health conditions of old age are actually related to some modifiable influences, governments around the world have implemented what are called healthy or active ageing policies. And it's thought that this type of approach will actually have an impact on the healthcare costs that might be associated with ageing populations. Our own National Strategy for an Ageing Society, which is almost a decade old now, pointed out that it will be important for older individuals and for our society and economy to have older people spend as much time as possible in their old age in good health. So this is very much the push that government have these days. They want us to be healthy in middle age, moving into old age as healthy as possible. So with that in mind, I want to talk a little bit about the types of studies that we've done in this area around healthy ageing. And our studies are done not just in Australia, but all around the world. And particularly, we have doing many of these studies in the Asia region, like China and Malaysia. So the first study that I'm going to talk about is a study that looked at how people understood ageing and healthy ageing. Because if we're going to design interventions to promote healthy ageing, we actually have to understand from an older person's viewpoint themselves what is important to them. The second study is a longitudinal study that we've been conducting where we've been interviewing older people in Melbourne for the last 16 years. So we have a very good idea of how health particularly has been important in terms of how they've aged. And the third study is the Happy Life Club, which I really like that name, and it's a healthy ageing intervention that we've been conducting in Australia and China. So I'm just going to briefly talk about those. I'm not going to bore you with lots of statistics. I'm really just going to cut to the chase about what are our major findings. So in terms of healthy ageing, we've asked a number of people questions like, how do you describe ageing? What are the most important things to have as you age? What does a healthy old age look like? How can you achieve a healthy old age? And we've conducted these interviews, as I've said, all over the world, and there's a remarkable degree of commonality across the answers to these sorts of questions. And the major themes I've listed here, and I'm wondering whether some of the people in the audience actually identify with some of these themes. One of them is living a simple life, and by that meaning a stress-free life as well. Particularly in the Asian countries, many people are concerned with having enough money to actually live out their old age and having companionship, social connectedness in old age. Older people want to continue with their everyday activities, and I think, again, we all identify with this. All of us want to continue being as we've always been, and a number of the respondents in our interviews talked about being able to be physically active, eating well, doing all the routine activities that they've always done during their life. The third theme that we've extracted from these interviews is contributing to society. And I think that's what I was really trying to get across earlier on when I said that we should not think of population ageing as a burden, because older people do and want to keep contributing to our society. And they see this as being able to maintain their independence and keep contributing just as they always have. And the last point really has come out more in our Asian studies, where social harmony is a big factor there. There's a great concern, particularly in countries like China, about having harmony in their family life, having low stress, not have to worry about their children. Now, I have a quote here from one of our Chinese participants, which I really like, and I use this quite a bit because I think it really captures the essence of all older people and how they see ageing. And I've got a few Chinese words in there, which I'll try to pronounce correctly. So this gentleman's a teacher who lives in the southern part of China. He's aged 76 and retired. So again, you can see here the importance of physical health. We do think that physical health is the most important, except that we all should have Lao Di, Lao Wo and Lao Ban. Lao Di means having some money. For example, an old man who lives in a rural area should also have income. So this is a very important part of ageing in poorer countries. Money is very important. Lao Wo means having a dwelling to live in. So obviously that's an important part of having a healthy life in old age. A senior also needs a Lao Ban, a partner, or else he's going to be lonely. If an old man or woman does not have a Lao Ban, he or she will be very lonely, and this will upset him. Therefore, apart from good health, we also need the 3 Laos. Furthermore, we need a cosy home and welfare. So by that, he's meaning we need to have a good income support to make sure that we can live well. And of course, the other areas that I've talked about already, ease of mind, meaning a stress-free life, a healthy diet, and physical activity. So this man, I think, has captured very well some of the areas that our older people that we've interviewed have talked about in terms of healthy ageing. And I've just got a little picture there of, this is a typical Chinese scene in a multi-generational family where we have about 4 generations of women who are living in the same area and in the same house, and that's one of their social activities. And I think it really captures that issue of having social connectedness in old age. In terms of our longitudinal study on healthy ageing, we've been following 1,000 older people in Melbourne for the last 16 years, and again asking them questions about their health. And we're particularly interested in what are the factors that predict healthy ageing in this particular cohort. So we've been following these people for 16 years. We've defined healthy ageing in a particular way. We asked them about how well they're doing it from a functional point of view. Are they independent in their functional abilities? Can they do everyday activities around their house? We also look at their psychological well-being, and we look at their general self-rated health to actually define a person who's ageing well. What we've found is that there are a number of factors that again equate with some of this interview work that I described earlier that are very important predictors of ageing well. So things like having few medical conditions or else having those medical conditions managed well, being physically active, having a healthy diet, not smoking, having good social support and good sleep patterns. And again, these are all factors that to some extent are within our control, but also we can design services that are going to actually promote these good types of habits. Lastly, I'd like to talk about an intervention that we're doing to promote healthy ageing in the community. And this particular intervention that we're doing is looking at how people manage their chronic illnesses. So if they have arthritis or diabetes or multiple chronic illnesses, how can we assist them to manage their chronic illnesses better? And this is very much what's called a client-centred approach. We try and give the older people skills for them to manage their illnesses in partnership with their doctors and nurses. And what we do is train up doctors and nurses to actually assist older people in managing their chronic illnesses, particularly around improving their health behaviours, such as physical activity, eating well, social activity. And the program is also designed to increase social activity within their communities. And as I mentioned earlier, we're actually running these programs both in Australia and in China. So what have we found with this study? We've actually found that we can decrease many of the risk factors associated with not ageing well. For example, we've shown that we can decrease blood sugar levels in those people with diabetes. We can decrease weight as well. We can increase protective factors such as good cholesterol. We can increase physical activity and improve their diet. We also have found that people who participate in these programs report improved quality of life, improved psychological well-being, and are much more confident in managing their own illnesses. So this approach in what's called a primary care setting, with it using doctors and nurses to assist older people to manage their illnesses, has been highly successful and has led to great improvements for the lives of these older people. So, in summary, what does that actually mean for us? I've said for you, but I mean for us as well, for me as well. What I'd like to conclude is that many of the health conditions of old age are actually associated with modifiable risk and protective factors. So when we look at those areas that I've mentioned and put them all together, I think one of the keys to having a healthy life in old age is to try and increase your physical activity, to have a good diet, not to smoke, moderate alcohol use, not abuse. We'll do that later. Good sleep habits, low stress, and I'm going to be interested in our last speaker who's going to talk a little bit about stress and cancer, which is going to be quite interesting. Well-managed chronic health conditions and social engagement. Now, a lot of these things we will have input to as individuals, but we also have to set up a society and an environment that is actually going to promote these healthy habits. So it's not just up to us as individuals to do this, but to make sure our health services are designed to achieve these sorts of outcomes, and also that our government policies are actually oriented to a more positive approach to ageing. So I would say healthy ageing is achievable, and that we have well-tested interventions in the community that can be applied. We know what are some of the modifiable risk factors that can improve our ageing experience. And I think really what we need to think about at the moment is how we're really going to push forward these approaches and have a much more positive approach to our ageing population. Thank you.
Thanks very much, Professor Browning. That's a fascinating topic, and I think it might make us all take a little bit more responsibility for ourselves and our own health as we age. Now please welcome to the stage Professor Peter Curry, who will speak about regenerative medicine.
Well, thank you for that. I guess as I'm the ageing male in the presentations tonight, and I know I'm going to get old and die of a disease, what I'm going to tell you about today is particularly pressing, and really what I want to give you is a snapshot of the research that's going on at the Regenerative Medicine Institute, which is one of the newer institutes at Monash University. And what we're trying to do is couple understanding of the lack of the regenerative capacity of the human body with some aspects of science about those organisms that can regenerate. So I'm just waiting for the slides to come up. And great. So most of you will be sitting in the audience, if you're like me, has some part of your anatomy that no longer functions like it used to. And this, of course, is the compelling reason why we're all sitting in the audience and why I'm here and why I do the things I do. So what we would ideally like to do is take all of us and those bits of us that don't work so well and regenerate them into new forms. And the concept of regeneration is actually a fairly ancient one. The ancients knew about regeneration. Prometheus had his liver regenerated when it was pecked out by the eagle. And in this rather striking image by Singer Sargent, you can see the Leonian Hydra, who had the rather unfortunate attribute when you chopped off its head, too, grew back in its place. So In mythology, regeneration has been considered a negative. And what I'm going to try and tell you today is there are some organisms in real life that do regeneration so much better than us, and deconstructing why they can do regeneration and why we can't is a very simple premise that we might find useful in unlocking our own regenerative potential. So in real life, the hydra itself is not such a scary organism. This is an invertebrate of the hydra class. And the hydra has the same potential as the Linnean hydra. If you cut it into small pieces, it will undergo a type of regeneration called epimorphic regeneration. That means the cells, each individual cell within this complex hydra, which has a nerve net and tentacles, as you can see, can regenerate the entire organism in toto. So that's a rather amazing ability, and you can see experiments listed there where this slice and diced hydra has regrown entirety of its organismal being. As you go up the evolutionary tree, you'll see other things that can do the similar things. So if you're like me, you've spent some time down on the beach and have the young boy propensity of pulling things apart, you would have pulled the arms off a starfish before and had your mother say, "How dare you do that?" And then if you'd come back a week later, you would have seen, in fact, that the arm would have regenerated and there was no need for it to be chastised. And there are other invertebrates, such as little planaria, which in this rather unfortunate experiment has been sliced from head to tail and allowed to regrow. And each of these little slices of the planaria can grow back into an entire organism. Now, this is an organism, again, that has a complex nerve net, it has a sensory system, it has organ systems, it has stem cells, many of which are similar to our own. And why is it that this beastie can do it and we can't? So if you look at the concept of regeneration broadly across the phylogenetic distribution of animals, here we are up the top, mammals, and our regenerative potential is in a few systems. I said, thank God we can regenerate our liver. If I was to choose one system we could regenerate, I would have had the liver, so thank you, Darwin, for making my liver regenerative. But there are other systems like the blood and the skin, which of course we know are regenerative. But if you go down to the other end of the system, there are a number of organ systems which cannot regenerate, and these actually are cause amongst the highest level of mortality. The fact that they can't regenerate is one of the highest reasons for morbidity in the Western world. So in my lab, we're very interested in exploring this concept of regenerative potential along phyla. And I'm interested in this particular organism. So this is the zebrafish, just the head of the zebrafish, and we're very much interested in most of the organ systems in this animal. It's perhaps a sad fact of life and a reflection that we are all just modified fish. We arose from fish evolutionarily, and there's nothing clear in the fossil record that we are basically modified fish. So many of the organ systems in the fish are very similar to our own. If you're interested in higher order brain function, perhaps it's not the system for you. Or if you're interested in placental interactions, it might not be the model. But it does model many behaviours and attributes of the human condition. And it's propelled this small little freshwater teleos species to the forefront of biomedical research. Why is that? Well, as I've said to you, zebrafish is basically a regenerative machine. Unlike the invertebrate systems, which often don't have very complex organ systems, fish have many complex organ systems that are similar to our own, but are highly regenerative. And they seem to be using the same systems across many organ systems to unlock their regenerative potential. And the other nice thing about zebrafish is that they are completely optically clear as larvae and embryos. So you can see all the organ systems forming. So in this little movie I'm running on the right here, it's the first 2 days of the zebrafish life. Just in a drop of water under a microscope, you'll see the embryo forming, the little muscle blocks forming. Here's the eye, the head, the tail. It starts to locomote. It hatches out of the chorion and liberates and swims away. And the ability to access the embryology of the system is a really important fundamental attribute of the system that we can greatly exploit to understand the mechanisms that are used to create these organ systems in the first place. And at the Monash University campus, for those of you who alumni have visited, you realise there's been a series of large buildings that have been put up on the biomedical space. This is a very exciting development, and it houses most of the research active faculty in the field of biomedical science. And in the basement of one of these buildings is a massive aquaria, in which we keep roughly 200,000 zebrafish in 9,000 tanks. And this ability to keep a large number of fish gives us fantastic genetic resolution and allows us to discover genes that are involved in the regenerative process. So here's a simple experiment, one you should not try at home, of course. Here is a fish where you simply cut off its tail, and a gene that's required for it, if it's knocked out, it doesn't regenerate. So that's a very simple-minded experiment to try and find out genes that are involved in regeneration of the tail. We're more interested in things that are fundamentally more close to problems that are associated with the human condition. So, of course, if we have a spinal cord injury that severs our spinal column, all motor activity below the severed point will be lost. And this is just a sad fact that leads to quadriplegia and paraplegia. However, if you do the same experiment, if you produce spinal cord injuries in a fish, this fish has had such an injury undertaken, you'll see that it is paralysed. But if you come back 3 weeks later, you'll see the fish basically has completely regained its motor function control. And in these panels up here, you'll see the replacement of the damaged nerve area and neurons crossing that site. And over time, these particular complex interactions and regeneration occurs. Now, we've been studying this for about 2 years, and we think we understand the basis of this. Of course, if we could understand why in this system, which has all the same cells that we do in our body, why in a zebrafish case it can fully regenerate its spinal cord and we cannot, then that would obviously be direct therapeutic relevance. And we've isolated a molecule that when we inject it now into the mice can actually increase spinal cord regeneration. It's a fish-specific molecule that seems to aid in its regenerative capacity. So that is one example for what I call the fish tank to bedside approach, and that molecule is currently in clinical trials. Another of our favourite organ system is the heart. Here we have the 3-day heart of a zebrafish embryo. It's a very simple organ system. It's only 2 chambers. We have 4 chambers in our heart. And if you look at, specifically look at our heart, now if you go quiet and listen specifically to your heartbeat at the moment, while you're watching this movie, it's much more effective. So the heart is a pumping machine. It's made up of striated muscle cells or cardiac muscle cells, which at their heart have this structure called the sarcomere. And the sarcomere is made up of protein structures which interact with each other and pull on different thread-like structures in the response to calcium. Basically, your cells are contracting in each heartbeat thousands and thousands and thousands of times a day. Now, it's an amazing thing that your heart even gets through the first week of life, let alone the first 70 years. But however, one of the major problems in the Western world is heart disease, brought on by many of the things that Colette talked about, and 60 to 70% of us will end up with some form of disease, and many of them will be coronary heart disease. So just to illustrate the problem a bit more dramatically, so heart failure occurs in around 300,000 individuals each year in Australia, and there are only HE transplants available. So our concept is, again, if we can understand this structure, which is the adult zebrafish heart, this 2-chambered structure, and work out why in this capacity it's able to regenerate, then we might have a better ability to regulate our own ability to produce regeneration in our own context. So as I said, the heart is made up of a series of different cell types. One of them is the myocardium, which is actually the muscle cell that contracts that I showed you in that movie. And in the myocardium, it's thought that they're a very low level of replacement in the adult heart of a human being. But however, the zebrafish response to injury is dramatically different. You can actually take the ventricle of an adult zebrafish, cut off 1/3 of its ventricle. Again, please don't try this at home. And you'll find within a month, the entirety of that resected area has regrown. And this is a remarkable concept. And I'll just show you a little bit why we think that's right. I'll talk you through that fact a little bit. So here's our unsuspecting zebrafish about to undergo heart resection. So as I said, the zebrafish have a very simple 2-chambered heart, and they also have a fairly simple circulatory system. So the arrow follows the blood that's pumped by the ventricle below here and the atrium above. So in the resection model that I've talked about, what happens is the steady hands of the surgeon comes in, resects roughly 1/3 of the ventricle in this particular case, and obviously no fish were hurt in the making of this film. And in this, you get some ensanguination of the blood. A clot forms, don't worry, everything will be fine. Now the question is that this is where we would stop. The clot would form, fibrin would form, collagen would form, there would be no recovery of any structure below that. And that would cause dilatation of the heart because the rest of the heart would have to function more fully. However, in the zebrafish, a number of very stereotypical things happen. The outside layer of the heart, called the epicardium, which is a very important pink tissue here, envelops the clot area. And actually what happens is a very amazing thing, that the heart cells that are at the wound site actually turn back into stem cell-like cells. They de-differentiate into what's a progenitor type cell, like a stem cell. They start to proliferate and fill the zone where the clot is. And really, one of the fundamental questions is, what are the triggers that make this cell type in the zebrafish turn back into a progenitor, regrow the damaged tissue, and what are the signals here emanating from those cell types that also trigger the pink stuff, the epicardium, to make new vasculature? And here are the signals coming, signaling to the cells in the epicardium, making the vasculature regrow, such that at the end of the day, you have more muscle and more vasculature, blood vessels filling that area. And how is it that signals that are generated in this context, how can we identify them? So one of the main issues that we're interested in trying to do is that, of course, in the mammalian context, you have an infarct caused by the block in a coronary artery in this particular case, and the area here, which lacks an oxygen supply, dies and never regrows. So if we could have magic fish juice, whack it on the heart, rub it in, make it regrow, then that situation would not be so life-threatening. So that's the basic concept of that branch of science. And some of the drug companies have cottoned onto this idea. Here's a very well-known heart drug, which is a lipid-sensing drug, and they reckon that we should look after our hearts because they can't regenerate like zebrafish. So you know that your system's made it when it's used in the publicity of a major drug company. Really interestingly, this concept probably sounds like a million miles apart. There's hundreds of millions of years of evolution. Maybe we've lost something vital. Maybe the trade-off is that having regenerative capacity is somehow harmful when you get to be a more complex organism. However, recent evidence suggests that that's not the case. So in the early mouse heart, mammalian tissue which can't regenerate as an adult, if you damage it, in the first couple of days of life after they're born, they are perfectly happy and able to regenerate. So this suggests that all the genes and the makeup and the complexity of the signalling is all there, but somehow in making a more complex adult homeostatic or an organ that needs to maintain its integrity in the adult, we lose that capacity. So it suggests that the idea is not so far-fetched, that we could tweak a few genes and turn ourselves into regenerating machines like the fish. I'm going to end my talk with a champion of regenerators. The fish is fantastic, but this little guy, newts, are amazing. So many of you have probably heard about Mexican axolotls. You've seen them in these little white guys in tanks and newts. But what you probably didn't realise, that if you as a child hacked one of the limbs off these guys, that it could regrow. Oops, sorry. So again, in these guys, they possess all the nerves, the bone, the muscles, the complexity that we have in our own arms. So how is it that when we resect a limb of an axolotl or a newt, this cute little guy here, that they can repopulate the entirety of their organ systems within the limb? So here he is again, unsuspecting surgical subject. The steady hand of the surgeon comes in again. There's an insanguination. No newts were hurt in the making of this film. The blastema, a special set of cells will form. So the wound occurs. Normally in us, if we lost a limb, there'd again be a clot and a wound would form, and then a scar that would never be resolved. But in the newt, they form this specialised structure, these little green cells called a blastema. Again, these are cells that turn themselves back into stem cells, and then are able to make white and red and green guys, all the different tissues that make up the limb, regrow and reform a limb. And the amazing thing is that those cells actually know where they came from. So if you do a resection higher up in the limb, it'll make the entirety of the limb. If you transplant that onto a resection that's further down, it'll still make the entirety of the limb. So it knows where it came from. So I'm going to leave my story there. There's our little friend here, who holds the secrets of unlocking our regenerative capacity, and I hope I've tried to engender some enthusiasm of studying these creatures and understanding why they undergo regeneration and we can't. Thank you.
What amazing, groundbreaking research. I think I'm a bit speechless. Now we welcome to the stage our final speaker for the evening, Dr. Erica Sloan, who's going to present on chronic stress. Dr. Sloan.
Thank you, Louise. I remember in first year university cutting up planaria. I'm not sure any of ours actually regrew, but we tried. So I'd like to talk to you today about our research on chronic stress and cancer. And I wanted to start with a personal story just to set the stage a little bit. A number of years ago, I moved to UCLA in Los Angeles for postdoctoral studies. And so I'd just finished up my PhD studies just a few blocks up the road, actually, at Peter MacCallum Cancer Centre. I'd spent 4 years studying genes that are involved in metastasis, which is a process whereby cancer cells move out of the primary tumour and colonise distant tissues. And it's this process that's responsible for much of the morbidity and pretty much all of the mortality of cancer. And so in the last month, I rushed around and I finished my thesis and I wrote up publications from the work and I packed up my house and I moved to a new country and I started a new job in a totally new field. And it was really exciting and it was just a little bit stressful as well. And so this was about mid-November and I knew that I just had to make it through to Christmas and then I could stop. My in-laws had invited us up to spend Christmas in Calgary, and on the 24th of December I woke up to a foot of snow and strep throat. And so Christmas was very memorable for all the wrong reasons. I felt like someone was putting razor blades down my throat every time I swallowed. And so you know the story, this probably sounds quite familiar, this idea that you're stressed but you've just got to get through to that deadline and then it'll all be okay. And then you get sick. And so we sort of accept this as part of everyday life. Stress is great, it motivates us, it gets us to that deadline. And we also kind of accept that hand in hand with this modern lifestyle is this possibility that it suppresses our immune system in some way and that we're more susceptible to infection by, for example, viruses. But what about cancer? That's the question that we asked in my lab. Is it possible for something like stress to impact a disease that's as chronic as cancer? It turns out the answer is yes. So the work from my lab over the last few years has shown that stress can indeed impact something as profound as cancer. 3 years ago, I was recruited back from Los Angeles to Monash Institute of Pharmaceutical Sciences, just up the road in Parkville. We use mouse models of human cancer to model the progression of cancer, to model that process of how cancer cells move out of a primary tumour and move out to distant sites and colonise those tissues and stop them working so well. And we use highly sensitive imaging technologies to actually track the process of stress as it occurs in real time. And so using these studies, we've found that chronic stress particularly impacts that process of metastasis, that process of progression where a cancer moves from a primary tumour out to distant tissues. And it's this way that stress can really impact the morbidity and the mortality of cancer. So how does it do it? How does stress accelerate cancer progression? Our research has found that stress acts on the tumour microenvironment and changes the environment within a tumour in a way that can help cancer cells escape. And surprisingly, it does this in a way by changing the specialised function of immune cells called macrophages. So you might have heard of macrophages. They're kind of like the border patrol cells in our body. They're surveillance cells. They go out through the body and they look for incursions, viruses and bacteria, and they protect us when there are viruses and bacteria in our body. When they sense them, they call in the heavy artillery, they call in T cells, and it's these cells that then do the killing of those pathogens. And then the macrophages step back in normally and they clean up the mess. But something goes wrong in cancer. So in cancer, these cells, these macrophages get subverted. And what we've found is that stress really helps change this process. So when you're stressed, these macrophages come into the tumour, but rather than being good macrophages and helping protect us, they stop their surveillance activity and they start to act like a fertiliser for the cancer cells. They help the cancer cells proliferate and they help them find a way out of the primary tumour and to disseminate to distant tissues. And we've found that they do this in several ways. One of the ways that they do it is they help grow new blood vessels into the tumour. And these blood vessels not only provide nutrients to the tumour, but they also provide a highway that allows the tumour cells to move out of the tumour and move to a distant tissue. So are we saying that stress causes cancer? No. Interestingly, our work hasn't shown any evidence that stress actually causes the onset of cancer. And this has also been sort of backed up by human studies that have been done where there's been no consistent link found between chronic stress and cancer onset. And so that's not to say that psychosocial risk factors don't affect cancer etiology, but rather to say that it's been very hard up till now to see consistent links between that. Although I'd like to come back to this at the end. In contrast, what we have found is that chronic stress really seems to affect this progression. And so this has implications for how we can use this information. In a way, this is sort of positive because it means that at the time of diagnosis, if you're sitting in a doctor's office and you've just been diagnosed with cancer, it means it's not too late to do something about it at that point. It means that at that point, there is still time, there's still a window of opportunity where we can look at how stress regulates cancer progression and we can think about ways to intervene in that and perhaps change the trajectory of cancer progression. So perhaps the all-important question, what sort of stress are we talking about? So as many of you will know, this is exam week at Monash University and a number of my undergrads think they know exactly what I'm talking about. But actually, acute stress can be highly motivating, both for getting good grades, but also for stimulating the immune system to protect us. In contrast, chronic stress or prolonged stress has been linked to really serious health consequences. And so what I'm talking about are things like major health stresses, death of a loved one, divorce, prolonged caregiving for a sick child or perhaps an ill parent. Ongoing situations where we feel like we have little control over what's going on. So that's fine, but how does stress actually get into our body to affect our health in this way? There are two main pathways that stress acts on. One is through the production of a hormone called cortisol that I'm sure you've heard of. This hormone is made by your adrenal gland and it affects your blood sugar metabolism to allow your body to be better prepared for a chronically stressful situation. Cortisol works by tending to suppress the immune system. The other pathway you probably know of is the fight or flight response. So this is when you're in a stressful situation, public speaking for example, and your palms get a bit sweaty and your heart starts to race. This is your sympathetic nervous system getting activated. This is a stress response that seems to be particularly important for cancer progression. The sympathetic nervous system kicks in and it makes neurotransmitters such as adrenaline and noradrenaline that bind to adrenergic receptors. So these are receptors present on the surface of cells such as cancer cells and the macrophages that are present within the tumour. And so binding to these receptors, the stress neurotransmitters can then change the behaviour of either the cancer cells or the immune cells in a way that helps the cancer cells move out of the tumour and spread to distant tissues. So that's nice. We know a bit about the pathways that stress uses to get into the body and we know a bit about what it actually does to the tumour environment. What can we do with this information? Well, it would be nice to say that we should all stop stressing. And actually there is evidence that if we can do something with our behaviour, if we can change our behaviour in a way that modulates our coping responses to an adverse situation, then we might be able to do something even in the context of cancer. So there are some really quite persuasive and quite well-controlled studies looking at populations that have shown that behavioural interventions can really change the trajectory of a cancer diagnosis. These studies have used things like cognitive behavioural stress management and mindfulness-based stress management to change how people view the situations. And these studies, surprisingly, have actually found differences in how the patients do and how frequently that process of metastasis occurs. Consistent with this, I actually became interested in this whole sort of field of study through my own practice of yoga. I took up yoga the same week I started my PhD, and to this day I'm completely convinced it's the only thing that actually got me through my graduate studies without having a complete breakdown. 14 years later, I still practice yoga sometimes, but I also know that as my stress levels increase, my self-care tends to go down and yoga goes out the window along with sleeping well and eating well. And so as much as I love the idea of telling someone with a cancer diagnosis that they need to not stress and they need to take up yoga or kickboxing or whatever suits your temperament, it's a great idea, but it may not work that well. And the other thing we know from these behavioural intervention studies is dose is really important. The more you practice, the more this will have an effect on your health, consistent with what you heard from Professor Browning. So what we've been investigating is how can we take this knowledge about how stress affects our bodies and design new drugs that target cancer by targeting the stress response. So now that we've identified the particular pathway that stress uses to get into the body, we can look at drugs that are actually in wide clinical use. We also learnt from Professor Browning that 37% of people over the age of 65 have incidents of hypertension. And one of the main drugs that's used to treat hypertension are beta blockers. What we wanted to know was, can we use beta blockers to block the effect of stress on cancer? So beta blockers will bind to the receptor that these stress neurotransmitters act through. And so we were curious to know whether that could also block the effect of stress. And this is exactly what we found. So in our mouse models, we see that when we treat mice with cancer who are very stressed, with beta blockers, we see a great improvement in outcome. And even more intriguingly, there's been a number of studies published in the last 12 months looking particularly at breast cancer patients who did not have breast cancer, were not diagnosed with it, they had hypertension and they were being treated with beta blockers. And then they received a diagnosis of breast cancer. Those patients who were being treated with beta blockers did much, much better than the other patients who were being treated for their hypertension with calcium channel blockers or ACE inhibitors. And so this is sort of quite persuasive evidence now from humans as well that these stress response pathways might play an important role in cancer progression in humans as well. So is it just breast cancer? To look at this, we've developed models of other tumour types, and it seems like this is a more general phenomenon. It seems like it affects other tumours in addition to breast cancer. We're starting to get evidence that pancreatic cancer might be influenced by stress response pathways. And we also have some evidence that leukemia, hematological malignancy, might also be sensitive to chronic stress. So where does that leave us? One thing I really like about this research is that it's so grounded in the reality of people's lives. I was in the US last week talking with a group of psychologists and epidemiologists and medical doctors about stress and cancer at an interdisciplinary meeting. And I came back through Los Angeles, and while I was there I stopped over and had lunch with a girlfriend on Saturday. And we were talking about how we were each doing, and she was telling me that she is Ashkenazi Jewish, and she had been thinking for a number of years about getting tests done for BRCA1 and BRCA2 mutations. So BRCA1 and BRCA2 are two genes that are involved in monitoring the integrity of our DNA. And when these genes get mutated, then our DNA, if it's damaged, is not properly repaired. And so this means it's much more likely that cancer will arise. It tends to lead to increased risk for breast and ovarian cancer in women and prostate cancer in men. And it's known that 1 in 30 people in the Ashkenazi Jewish population carry this mutation, and so are much more likely to be diagnosed with cancer. And so about a year ago, she had got up courage to be tested for these mutations and had found out that she was indeed a carrier for it, which meant that by the age of 40, she would have a 20% risk of having breast cancer. And so we had lunch on Saturday, and in addition to giving me permission to share her story, she told me that she had decided to take action. Last week, she had a prophylactic double mastectomy. She also said that in a couple of years' time, she'll consider having a prophylactic ovarectomy to further protect her against the possibility of ovarian cancer. And as she commented, if you're going to get breast implants, what better place to do it than Los Angeles? So this is great. This is something proactive that she's done that will reduce her risk of developing cancer. But we also spent quite a lot of time talking about the stress both involved in deciding to get the mutation analysis done, the stress of deciding what to do with that information. And it made me think that even though we haven't been able to find links between stress and the onset of cancer up till now, perhaps we're looking at the wrong population. And so our next study is clear. We clearly need to look at how stress impacts women who've inherited a greater risk of cancer. And so that's what I really love about this work, that stress is with us, it's part of us, it's part of our modern society. We thrive on it and it motivates us. But unmoderated, it's also harming us. And so each conversation that we have with someone with a personal experience of cancer helps us to refine our thinking about how stress signals into the body and affects our health and what we can do about it. And so thank you.
Thank you, Dr. Sloan. I think that's a very clear message about why managing our stress and reducing it if we can is imperative to our healthy living. Tonight we've showcased just a snapshot of the great research Monash academics are undertaking in an effort to improve our lives. There's really exciting research happening at the university and we're very proud of the achievements. I'm also feeling a lot better about ageing and illness as we get older. Please join me again in thanking our speakers tonight for their thought-provoking presentations.